Hello, everyone. Thank you for joining us today. I am Yamina from Dr. GPCR, and today's session is presented in collaboration with our partners at Eurofins DiscoverX, the industry standard for GPCR cell-based assays with over 25 years of expertise and billions of data points across thousands of peer-reviewed publications. Our guest and speaker today is Dr. Terry Kenakin. He's professor of pharmacology at UNC Chapel Hill. He has over 40 years of foundational work in receptor pharmacology and is also heading Terry's Pharmacology Corner here with Dr. GPCR. Quick housekeeping items. This session is being recorded. Please keep your microphones muted during the presentation. At the end of the presentation, we will open the floor for questions. If you prefer not to appear in the recording, just feel free to type your questions in the chat and I will read them. If you are comfortable being on the recording and asking your questions, you'll only have to raise your hand or just jump on. I'll call on your name and you'll be able to ask Terry your questions. And with this, Terry, the floor is yours.
Thank you. Hi, folks. Thank you for coming. Thank you for your interest. I'm going to share my entire screen and get my slides up, and we will begin. And as Yamina said, probably better to do questions at the end, and I think we'll have enough time to program. And so what we will talk about today, we'll start with defining what we mean by ligand bias and what causes it. And then I'll talk about how to use it in your program, how to know it when you see it. And the whole key here is assays. What I'll tell you is bias is just standard protein allostery, but we didn't really understand that until we had the assays that separately looked at signaling. Then it was obvious that compounds that produced response did not produce it in a uniform way. And so we'll talk about some assays. And our co-hosts here, Eurofins DiscoverX, are a great source of assays there, and so I'll talk about those a bit. And then finish with how to quantify it as a property of your scaffold. And this is really taken on mythic proportions with methods and equations and so forth, which we don't need. It's a very, very simple thing, and that's what I hope to convey. So let's start with some ground rules. Bias is a natural outcome of allostery. All proteins do it. So what I'd like to impress upon you is it's happening in your program right now, no matter if you seek it or not. So to ignore it is really walking away from some potentially something of great value. What do I mean by it's inherent to allostery? Let's look at panel A. We have a protein and it's interacting with two tracers, tracer 1, tracer 2, and we add an allosteric compound. That changes the shape of the protein. Proteins, of course, are extremely complex, and a change in shape does not mean you will have an equal effect for both tracer 1 and tracer 2. In fact, the evidence is you rarely ever do. This is what is called allosteric probe dependence. So my allosteric compound has affected tracer 1. It didn't do that much for tracer 2. Well, if I take those two tracer sites and I put them inside the cell and give them names and call them G protein site and beta-arrestin site, that is all bias is. It's functional allosteric probe dependence aimed towards the cell of signaling systems, and it's happening all the time. So if asked, should we seek it in our programs? No need. Bias will seek you. So when you look, I do not know at present, and it's hard to say if this will change, but I don't know of a target that does not practice bias. So we have small molecules, a whole host of those. It occurs with peptide agonists. This is GLP-1 tirzepatide, peptide antagonists. antibodies, and at the end I'll mention all allosteric compounds induce bias, and that's a very important point, perhaps of interest in a later webinar. So back to our ground rules. Bias is occurring. You should not ignore it. I'm often asked, well, what if I'm not interested in G protein versus beta-arrestin? What if I'm interested in other pathways? That isn't the point of a bias assay. Bias assays will identify agonists that make a unique active state. So you're basically using it as a surrogate. That does not mean that you have to stick with the assays you've used. For example, GS protein beta-arrestin. It simply picks out the compound, and then you would put it in your natural system, and knowing that it makes an active state that's unique, see what that does. So no need to worry about what pathway you would have an interest in. Just get two separate ones. The most common ones, of course, are G protein beta-arrestin. And then we'll talk about how we could exploit this for therapies, and finally how to detect it and quantify it. In fact, I'm going to start with that. So very important to have a toolkit, and there are many new ones. It was quite simple when I was a student eons ago. In the Ice Age, you had cyclic AMP, calcium binding. Now we have a plethora of very exotic assays, and they're extremely detailed, textured, and they're the key to determining what our scaffold is doing, because our scaffold is giving a fingerprint of efficacies to the cell, and many of these are different. GPCRs interact with a range of signaling. They have a choice, and that's what we will be talking about. When I talk about efficacy, it used to be a functional readout. Really, I mean now a much broader range of that. It's the property of your scaffold that when it binds to the receptor will change the behavior of that receptor towards its host. That'll be the cell. So it's beyond just signaling pathways. Of course, you have cyclic AMP, IP1 calcium, beta-arrestin, but also what the receptor does with the cell. It internalizes and does all kinds of things. All of these are efficacies that we can now try to exploit therapeutically. So let's get on to some of these gorgeous assays that we now have. And I won't go into them in detail. If you want to know more, go on the Eurofins DiscoverX websites. They're beautifully explained with examples and so forth. But we have cyclic AMP with Hit Hunter, calcium, and this calcium no wash assay. What else? Beta-arrestin interactions, ways to see the interaction of receptors with G proteins, and even beyond that. Where does the receptor go? Into the ER? And if so, there's ways you can explore if that leads to signaling, recycling, and so forth, and chaperones as well. So the point is, you have a rich array of possible options, and use these as tools to detect the specialness of your scaffold. And by that specialness, the fingerprint of the efficacy. Years ago, efficacy was considered as a single signal, and very in strength. It had quantity. Now we know, now efficacy has quality, bit of GS, bit of GI, and so forth. What you see here is 16 opioid agonists. These were run at Monash in 6 assays. And what we did was we took data output and clustered it. And we used the cluster program from Harvard called Morpheus, which is designed to cluster genes, but it'll take anything and say what is similar to what. And so we gave it this, and this is what it did. This is the cell telling us the way it sees agonists as being similar, not us with structure. Usually we would use structure. For example, the cells tell us that morphine, dynorphin A really produce similar signaling, yet structurally they're apples and oranges. So here's another way that we can see choosing textures in efficacy to give us therapeutic nuances. So what is this thing called bias and what causes it? The really first well-worked out molecule that was biased was Travena 120, and it's an angiotensin ligand. If you give angiotensin to its receptor, it will activate GQ protein beta-arrestin. Travena made an analog called TRV 120.027, binds to the same receptor, but only activates beta-arrestin, does not produce GQ protein activation. That's what we mean by bias, and that's what we hope to harness therapeutically. It's a very powerful tool, and we'll explain why. What causes it? These are those unique receptor states that different ligands stabilize. Receptors can form a whole host of these in an ensemble. Ligands come in there and stabilize certain subsets. So we have agonist 1, and maybe it makes a state that prefers GI1, G1. A different agonist may make a different state that will interact with G2 beta-arrestin. That's all bias is. Your assay is telling you that this compound is making some unique state compared to a standard, to the natural hormone, and can you exploit that therapeutically? So how do we use it in our programs? Well, of course, we all know about extracellular selectivity. We have an agonist and we wish it to signal to only a certain subset of receptors. Well, then it goes beyond that. Once it does that, there's another subset. We can now emphasize the outcomes in terms of signaling. And this is the whole new world that has entered the field for about 20 years now, at least. So let me just generally talk about the ways it has been applied. You can have a proactive approach. For example, you can emphasize a good signal that you want. Beta-arrestin 2 for PTH has been reported to possibly be useful for osteoporosis. So you may want an agonist that is biased towards that signal for that outcome. GLP-1, same thing. And there are a host of ideas out there. How can we emphasize a good signal? So that's one way. Second way is de-emphasize something you don't want. We all know that opioids depress breathing. This is data from Laura Bond showing that morphine produces respiratory depression, as we all know, but much less so in a beta-arrestin knockout mouse. So that would suggest if you wanted an analgesic that did not produce respiratory depression, you make it biased towards the G protein. And later we'll talk about some splendid data from Kate White on other aspects of that. Or we can de-emphasize something bad that the natural system does. And this is the the angiotensin vasoconstriction that is a natural reflex when you lose cardiac output. When your organs don't perfuse as well, you will get triggering of the renin system, release of angiotensin, pressure goes up, and everything's great in a healthy heart. In an unhealthy heart, is terrible. So what Travena did is said, we don't want that vasoconstriction because that'll make an unhealthy heart, make it harder for it to empty. But we do want the good beta-arrestin effects of angiotensin, so that's why they made Travena 120. And finally, back to Kate White. Kate showed if you think about kappa opioids, they're involved in a whole bunch of things: cognition, reward, mood, perception. So you could foresee some pretty good drugs there, except when you activate this target, you get dysphoria. Everybody knows about salvia. You get terrifying hallucinations. So it precludes this target from ever being pursued therapeutically. Kate showed that bad stuff was all beta-arrestin mediated. So again, if you had a biased compound that was biased away from that, you might open up this target as a potentially useful therapeutic one. So those are the proactive approaches. There's also retroactive, and this is commonly used because it's easy. So let's take some history here. We have a screening assay, and activity is going to the right here. So I've screened my compounds, and I get a few hits here. The convention has been, I'll take my most active hits, and I'll move on to an animal model or some other assay. Now it's realized that all these hits may not all be equal. And so you do cross-screening. You will rescreen your hits in a different assay. I guarantee you will get a scattergram. You will not get a correlation because of bias. So now what happens? Instead of going with my most active compounds, which all may be doing the same thing, I can take exemplars, that compound, this one, and this one, which I know are making different active states, and I put them to the more complex assay because the chances that I'll see a unique phenotype from those are probably much higher than if I'd have chosen just some random but active subset. These may be doing all the same thing. So you cross-screen, pick out exemplars, and you move those forward. Okay, so how do we see bias? There's a lot of models out there, equations, and it's been debated and so forth, but you don't need a model, you don't need an equation. It's very, very easy. The first step is you just construct a bias plot, which is here we have an agonist producing signal 1, signal 2. I simply re-express the signals as functions of the same concentration and make what is called a bias plot. So now I have the responses of this compound in pathway 2, pathway 1, plotted as a function of responses in pathway 2. And it'll be curved simply because these are two assays of differing sensitivity and so forth. So if I do two agonists now, and they're doing the same thing, I will get the same curvature. This is system bias. This is not something you could use because it's unique to the assays you've chosen. However, if you see this happening, departure of the bias plots. This is telling you something real. This will be carried on in every cell type you study. So what you are trying to see is a departure of your bias plot. That's once you have that, that's a compound which you might want to quantify, but in any case, you've identified as being biased. For example, here we have some data for new opioids. These are agonists, and you can see the bias plots for G protein activation, beta-arrestin for morphine, fentanyl, bingo, are pretty uniform. They're all probably doing the same thing, but carfentanil isn't. It's departed. That's the unique compound, and so you can pick it out and study it further. So it's pretty simple to see bias when it's presented. And this is some data from Eurofins DiscoverX, cyclic AMP in solid lines, beta-arrestin in dotted lines. So all you need are two curves. Here's the bias plot, and it quickly picks out chemokines that are doing different things. This is the kind of data we were using for our HIV program, trying to see compounds that would selectively internalize CR5. And finally, so you've identified your compound. Now it's useful for the chemists to have a scale. How bias is this scaffold versus a different scaffold? So let's talk about how to quantify bias. One way to do it, and we've used this and it seems to work well, is to apply the operational model, which of course will quantify affinity and efficacy, but you just separate it. You now have separate assays. So here I have a G1 readout, and I can quantify the efficacy and affinity of the compound for activation of G1, and I can choose a different readout, beta-arrestin, and again, the efficacy and affinity for the secondary pathway. Affinities don't vary much, but efficacies do. And so now we can start quantifying the relative efficacy for each pathway, and we can give that a number. But it's a little more complicated than that because of what cells do to agonist response. So what I've shown here is a weak agonist, and it's run in a magic cell. You can induce receptors and increase them, just dial them up. Your curves will do this. Here is in a low expression system, higher. But the point of this curve is that there's no uniform changes. It will do one thing when the compounds are partial agonists and a different thing when they're full agonists. And so really you have separate worlds. You cannot, it's difficult to compare a full agonist to a partial agonist, which is the main reason Black and Left created the operational model. Before them, you could not compare these. This model allows you to compare full and partial agonists through various numbers, and the number that I will talk about is a transducer ratio. So let's go through an example. Very, very simple. There's 4 ways to work out bias. Number 1, of course, you need a curve for each agonist. And you must have curves, otherwise you can't do it. You will see reports of compounds that are perfect bias. That is, they make responses in one system and no response in this one. Well, you're probably right. it is biased, but you can't quantify it. You must have a curve for each. So here again, we have DiscoverX, Eurofins data, Exendin, and two other GLP-1 agonists, and you should get each curve, quantify maximal response and an EC50. Maximal response is expressed as the fraction of the total window of the assay, so it cannot go above 1. So here we are. Step 1, we have that. Step 2 is we calculate what is called the transducer ratio. You simply work out log of maximal response divided by the EC50. And so I've just divided that by that, took its log, and these are extremely useful and powerful numbers because they reduce agonism to a single number that will quantify the power of each compound to activate that one pathway. So now I can compare partial agonists to a full because I have that index. So step 2 is we calculate this. Step 3 is to rank within each pathway the relative power of the agonist. We still can't compare across because these are different cell lines with different sensitivities and so forth. We have to correct for that. So we choose a reference compound. In this case, I've chosen Exendin 4. And I said, relative to Exendin 4, what is the relative power of these other two agonists? And so you just calculate delta log max over EC50, and there it is for each pathway. Now I've normalized for G proteins, beta-arrestin, according to a common reference. Now I can cross assays and compare what I'll call bias, and that's doing it again, delta log max over EC50, and there they are. Anti-log is therefore the bias, and so in this case, these two compounds are 17-fold bias towards cyclic AMP versus beta. And it's as easy as that. All it is you need curves and an Excel sheet, and you've got it. Bias is a selectivity, but it is different from receptor selectivity, which is you won't get an effect until you attain a concentration that begins to interact with this secondary receptor. So you will get no effects on pathway B until you get to the concentrations that you need to activate the receptor mediating that. Bias is not the same thing. Bias begins everything at once. You will get pathways activated, both A and B, the instant that the compound interacts with the receptor. But while they're activated simultaneously, they will be at very different volume strengths. So let me try to illustrate this. You will see data presented for bias. I just showed you this one, cyclic AMP, GLP-1, beta-arrestin. Let's take a compound and put it on the same concentration axis. This is tirzepatide. So all I did was I took that curve, put it here, took that curve. Now, does this really mean that I'm not going to activate beta-arrestin until I get to concentrations 10,000 times needed for cyclic AMP? No, that does not add up with mass action. What's happening here is cyclic AMP assays are very much more sensitive than beta-arrestin assays. So I can't just simply cross over. That's why I did that normalized to a standard within each assay. Then I can cross over. So what does that mean for if I'm interested in the receptor selectivity of GLP-1 for GLP-1 versus GIP, data shows us that GLP-1 curve is down here. GIP is way thousands of fold higher. This is receptor selectivity. If I want to know about bias within one of those, GLP-1, this is real data. And I apologize, I don't have the agonist here. This shows that the cyclic AMP curve is here, and all the other things are kicking in, but at a very, very much weaker rate, so you basically don't see them. You will still see a selectivity, but just know that everything starts at once. It's not a question of something starts and then you have to go to higher concentration before the second something starts. All this is good. I've hopefully shown you it's easy to do, it's easy to detect, quantify it. The issue now in the field is translation. We have these fingerprints, but now the compound's going to interact with a myriad of cell types and pathways, stoichiometries of things, and that's where the issue gets to be tricky. And so this guy has the relevant question. It's all well and good. I have a compound, but will it make him feel better? And I suspect we already have had a few cases of this, that biased compounds that are beautiful in vitro systems fall into hard rocks of in vivo translation. So this is where the field is now, which is why I said, once you have your scaffold that you know makes separate active states, probably get it in your natural system as early as you can to see if that plays out to be something useful. And I liken this process of translation to, if you read about, there was a coding cipher machine called Enigma that was used in World War II. And it was very difficult to decode, because what you would do is you would key in your message here, and it would go to a plug board of various options and get scrambled here, and then get further scrambled to 5 wheels that rotated, and then was played back out through a different rotation, different plug board to give what the coded message was. I just show this, other than it being interesting, it has an amazing number of possibilities. Well, in vivo, we're sort of facing the same thing with different stoichiometries of couplers, receptors, cell types, and so forth. So this is what we have to deal with to try to turn biased compounds into useful therapeutic drugs. I'll just finish with this. I mentioned it at the start. We're, of course, in an allosteric world now. GPCRs are nature's prototype allosteric protein. Everything this thing does is allosteric. So remember when I said any change in shape, any allosteric change in shape has the potential to be probe dependent. So if you're making an allosteric compound, PAMs, NAMs, they are changing shape. They are going to induce bias. You can't escape it. So separately, while you're interested in quantifying the allosteric effects, you now have to check, okay, what have I done to my natural hormone fingerprint when I did this? And there are many examples now of NAMs and PAMs that completely change the nature of the efficacy that they are aimed at changing. This is a potentially important topic, and I'm hoping that Dr. GPCR can explore it in future sessions. So I'll stop here. That's my story. If you want to read more, Eurofins DiscoverX has published a white paper on this, basically saying what I've said, but in much more detailed with evidence that you could read about. So, to recap, bias is not an option. It's happening already, so you may as well consider it. Moreover, there are many ways you could see it being exploited for therapeutic advantage, strengthening good signals, reducing harmful ones. But the key in all of this is having assays. That he or she who has the assay to see something that others cannot see has a definitive advantage. So I think that takes us to time, and I'll stop at this point and ask if anyone has any questions.
Thank you, Terry. We already have a question in the chat. So Ivana says, thanks for a great talk. In terms of bias quantification, what bias factor would be statistically relevant and which error is used to determine statistical relevance? That's number one. Then the chat continues, keeping in mind that low intrinsic activity ligands might show higher errors, which in turn complicates statistical analysis. Thank you.
Very nice question. Very insightful. The key is, in fact, statistics. When we published the operational method, we worked with a statistician.
Terry, I apologize for interrupting you. Can you please stop sharing your entire screen?
Okay. So, yeah, right.
There you go. Thank you.
Sorry about that. And so Steve worked out a way to use variance and statistically tell you whether small differences, sometimes you'll see biases of 2.5-fold, 5-fold. Is that real or noise? That pretty well takes care of the question of weak compounds, strong ones. You let the data tell you, okay, it's real or it isn't. So that one's easy. The harder one is, okay, yeah, but what's therapeutically relevant? How much bias do I need to change a system? And that is still an open question and will probably change from target to target. For example, GLP-1, we were trying to make a PAM that would potentiate GLP-1 to get an increased insulin release. One other way to do this is to inhibit the enzyme that destroys GLP-1. And there are those out there, and they produce about a 3-fold increase in GLP-1 levels. And that was enough. That was effective in diabetes. So there we had an outside answer. Bias I would need is about 3-fold. I don't need more than that. You don't often have that external cheat sheet that says this is how much I need. Bias differences usually aren't huge. You can get biases of 100-fold and so forth, but my guess is most physiology operates in the steep part of a curve where the system responds to the hormone and so forth. So a 5-fold difference is huge. The system is seeing a 5-fold increase in strength of signal. So I don't think you'll need these whoppingly huge 100-fold differences, but I don't know. I think it will vary from system to system. So I hope that covered that one.
Thank you, Terry. Anyone else? You can feel free to raise your hand and I will call upon you. Chris, go ahead.
Hey, Chris. Hi. Yeah.
Hi, Terry. Thank you again so much for your wonderful papers and your textbooks and now webinars. This is wonderful. And
my
really,
pleasure.
I was going to ask you, you focused in your talk today about G protein signaling versus beta-arrestin signaling and assays, which have now been really nicely available and quite reliable. Can you speak about the value of some of these newer assays that are measuring things like internalization, and maybe comment on their potential value for therapeutic program. I know it's an area of basic research and we don't necessarily have great excellent examples yet, but any insights you can share without disclosing obviously confidential information?
For sure. This thing opened up because of the easy, low-hanging fruit of G protein beta-arrestin. Now we have all these newer assays. There's no reason these can't be used every bit as well. And I think Dr. GPCR had Sudar talk about location bias. He's an expert in this field, where you're actually shuttling the receptor inside the cell, the various regions of the cell. It's doing different things when it happens. For example, MC4, this is a huge part of its repertoire. So yeah, all of these become important. But again, if you can get to what you think is your therapeutically relevant bias set of assays, perfect. But you really don't have to. You can use any one of those to say this compound is making a different state. Then let me explore it further. But yeah, way beyond that, there's another assay called QPath that was developed by Reed Olson and Jeff DeBerto. You can tell instantly 21 G protein interactions right away. I mean, it's amazing. So,
thank you, Chris. Anyone else with any questions? Aurelia, go ahead.
Aurelia, hi.
Hi, Terry. Thanks a lot for the talk. Very nice to listen to you again, Terry. It was really interesting that you described the bias as fundamentally allostery effect, right? And so, if I understand correctly, it's also true for orthosteric ligand, right? That it's binding orthosterically and it has a distant effect on another site, like the G protein site, right? Yes, that it's in that sense, it's allosteric effect. But then, when thinking about it, does it mean that if we compare allosteric ligand and orthosteric ligand, they would have the same propensity or same probability in generating bias, or would that be different?
Great question. Yeah. And here I'm going to cite some just some beautiful work that was done at Monash by Arthur Christopoulos and I forget the other co-authors. I apologize, but they were looking at the muscarinic agonists, and they were interested in cognition for schizophrenia and so forth. And it really fell out beautifully. They had orthosteric agonists and allosteric ones, and they separated out perfectly. This group was biased towards G protein, and this group was biased towards beta-arrestin. No, ERK, I think, but it was different, and it separated. And so one has to think that had something to do with where the compound bound, causing different changes in the ensemble to give you a different interaction with the cell. But in the end, these ideas of orthosteric and allosteric when it comes to agonism are a little bit redundant, because anything that binds, whether it's to the orthosteric site or an allosteric site, is going to allosterically change that protein. So we say binding to an orthosteric site, but it's producing an allosteric efficacy.
Thank you, Aurelien. We have time for maybe 2 more questions. Any other questions? I'm not seeing anything in the chat. My apologies.
If anyone has a question that occurs to them later, you can feel free to email me as well, and I'd be happy to take it up that way.
Absolutely. And I'm going to shamelessly plug here Terry's Corner and the free newsletter for people who want to continue reading on the topic and get a better sense of what are the topics that are covered in Terry's Corner. I'm going to put the registration link, sign up link in the chat. We still have a couple of more minutes. Please let us know if you have any questions.
Hi. All right.
I don't see any other questions in the chat, but wanted to mention that we are almost at the end. If no other questions are on, I have a couple, but Terry, I'm gonna leave those for another time, since I'm the one reviewing and watching all of these fascinating recordings. Every time you prepare something, I'm always excited to read about it and watch those. So if there's no more questions, I want to thank everyone for joining us today. Thank you to our friends and partners at Eurofins. Chris, you turned on your camera again, so I'm wondering if you have another comment to chime in with.
Oh no, I just look forward to more allostery discussions in the future.
We look forward to having you all, and we are already preparing our next webinar in September. We'll have more information regarding that one, again with Terry and our friends and partners at Eurofins DiscoverX. Thank you all for joining us. Thank you all for being here today. Terry, thank you again for the fantastic talk. Please don't go anywhere, Terry. Thanks, everybody. We'll see you again soon. Bye-bye.
Thanks, folks.
Thanks,
Bye.
Serge.